Hello and welcome. Today let's move further through this chapter and get chapter 7 taken care of, or our sensory physiology taken care of. Now when we go through, we talk photoreceptors and phototransduction is where we are at now from what we have left off from. Now I'd like you to know here we're going to get into talking about our rods and cones, our photoreceptors. So we'll talk photoreceptors and then we'll move into phototransduction, basically this explanation of how light is going to excite the photoreceptors and then help spread signals off to the brain. So we'll see once light is focused on the retina, that's when then the photoreceptors, our rods and cones, again, will come into play. So if we move back a few pictures and we come back here and we look at this picture right in here, we can see when light makes its way into the eye, we said light is going to cross the cornea, make its way through then the aqueous humor, through this pupil, passing through the lens, and then making its way all the way to the back of the eye. So let's look at this area here and what happens when light makes its way in that area. So here we could see, so light makes its way all the way through. You can see here it's going to come and it's going to hit that choroid and then our pigmented layer. And we said that their job is going to be to help keep that light from scattering, which would lead to then a distorted image, a blur, basically. Here now we can see in greater detail the pathway of light. So light is coming in this way. So it comes in all the way to excite the pigmented layer. Then here you can see our photoreceptors, our rods and cones, are embedded within that pigmented layer. So when we talk phototransduction, you'll see this could be one of your possible essays where you have to go through and explain the whole process. To sum it up in one sentence for you, phototransduction, you'll see, is the process by which light energy is converted into a graded receptor potential. So you have to be able to explain that whole process. Again, it doesn't happen in one sentence. I'm just giving you a quick summary there of what this process means, and now we'll go through and we'll talk about this whole process. So we've kind of already started when we talked about light making its way all the way through to excite then the photoreceptors is where we're at. So let's talk about the functional anatomy of these photoreceptors. So here, let's come back to this slide. Right inside of here. Now, when we talk about our photoreceptors are modified neurons. Our photoreceptors are modified neurons that are turned upside down, with their tips immersed into the pigmented layer of the retina. And you can see that here very well. So when we talk about the outer segments, here you can see the outer segments or the tips. They're immersed into the pigmented layer, we said. The tips are the receptive regions of our rods and cones. They are the receptive regions of our rods and cones. In rods, the outer segment is slender and rod shaped. The cones, they have a short conical outer segment versus our slender rod-shaped outer segment of the rod. The outer segments are important because they contain photopigments. Which are found in stacked layers of membranes called discs, So let's move into the visual pigments, the photopigments. So when we talk about our visual pigments, our photopigments, they're going to be found in the outer segments, and they change shape as they absorb light. We'll look at that. These pigments we'll see are going to be embedded in discs. They're embedded in discs, and you can see those discs are found inside these outer segments. So these pigments are embedded in discs and they help to trap light. Here we can see rhodopsin. Rhodopsin is the visual pigment in rods that we're going to look at. Here, this figure shows us how it looks. So moving back then. Next then we have our inner segments. These inner segments are going to be found located in between the cell body of the photoreceptor and its outer segment, found in between those two regions. And they're going to contain mitochondria, as we can see. They contain mitochondria and other organelles. Let's compare rod and cone vision now. Let's talk about the difference between rods and cones. Now first, our photoreceptor cells are vulnerable to damage. They will degenerate if the retina becomes detached, and the retina is very fragile. These photoreceptor cells can also be destroyed by intense light. The outer segment is renewed every 24 hours. Tips are going to fragment off and they're eventually phagocytized. So let's talk about our rods. I'd like you to know our rods are going to be very sensitive. That means that they respond to very dim light, making them best suited for night vision and peripheral vision. So we'll see in dim light, only the highly sensitive rods are going to be able to respond. Though rods are activated over a range of wavelengths that overlap with those that activate cones, there is no mechanism for distinguishing between frequencies, making objects that appear vividly colored in bright light appear in shades of gray as night falls. Our rods contain a single kind of visual pigment, so their inputs are perceived only in gray tones. So next time when we talk about our cones, our cones, they need bright light for activation. They have very low sensitivity, but they react more actively. The human retinas have three kinds of cones. One responding optimally at long wavelengths, our red cones. One that responds optimally at medium wavelengths, our green cones. And third, our blue cones, which are going to be stimulated at short wavelengths. Will provide us with a colored view of the world. And then last, we have color blindness, in which the patient will lack one or more of the cones. So next then here we can compare our rods and cones. Right, color vision, non-color vision. We talked about high sensitivity, low sensitivity. And here you can see also when we talk quantity, you saw way more rods. Okay, way more rods than cones. You can see that here in this picture. So rods are more numerous in quantity as well compared to our cones. Next one, we'll move to the visual pigment and we'll look at its chemistry. So let's now talk about the chemistry of our visual pigments. Here we will see that photoreceptors translate incoming light into electrical signals. But before we do that, I'd like you to know about retinol. Let's talk about retinol. Retinol is a light-absorbing molecule that combines with proteins, opsins, to help form four visual pigments. Retinol, it's important that you know, is going to be made from vitamin A. It's synthesized from vitamin A. The pigmented layer is responsible for absorbing vitamin A and keeping it there for the rods and the cones. Retinol can be found in a few forms. The first form I'd like you to know about is going to be 11-cis retinol. Now, when we talk about 11-cis retinol, 11-cis retinol is going to be retinol that's going to be bound to opsin. And 11-cis retinol is going to have a bent or a kinked shape to it. So we can appreciate that then. Here we've got our rhodopsin. So we focus in on that rhodopsin in greater detail now. Here we can appreciate our 11-cis retinol. So it has a kinked or a bent shape to it. Next, then we have our all-trans retinol. Now, our all-trans retinol is formed once light hits our visual pigment. The capture of light by visual pigments initiates a chain of chemical and electrical reactions in our rods and our cones that ultimately cause electrical potentials or electrical impulses to be transmitted along the optic nerve. So let's look at these events closely now. Phototransduction is the process we said by which light energy is going to be converted into a graded receptor potential, and it begins when a visual pigment captures light. So when we talk capturing light, again, the visual pigment of rods is this purple pigment called rhodopsin. So here we're going to look at the formation and the breakdown of rhodopsin. So first let's talk pigment synthesis. Now, rhodopsin forms and it accumulates in the dark. In the dark. Vitamin A, we can see, gets oxidized to form 11-cis retinol, which is combined with opsin to form then rhodopsin, as we see here. Next, then let's talk pigment bleaching. When rhodopsin absorbs light, it changes shape and it becomes all trans retinol. Light absorption by rhodopsin is going to trigger a rapid series of steps in which retinol changes shape and eventually releases from opsin, as we can see right down inside of here. This breakdown of rhodopsin to retinol and opsin, this is known as bleaching of the pigment. This is known as the bleaching of the pigment. Next then we can look at pigment regeneration. When we talk pigment regeneration, I'd like you to know once the all-trans retinol detaches from opsin, it is converted back to 11-cis retinol. Rhodopsin is regenerated when 11-cis retinol is rejoined to opsin. Enzymes are going to slowly convert all trans retinol to its 11-cis form in the pigmented epithelium. And this process will require ATP. So here we can see light activated rhodopsin activates, or we can say it gives energy to stimulate a G protein we see right inside of here that's called transducin. So once light hits the visual pigment, its shape changes, we said, right? We said it becomes all trans retinol. So our transducin, our G protein is activated. Transducin then activates phosphodiesterase, PDE, it's abbreviated as. Phosphodiesterase then, this enzyme, it breaks down cyclic GMP to GMP. In the dark, guanylyl cyclase converts GTP to cyclic GMP. Cyclic GMP then binds to cation channels, holding them open and allowing sodium and calcium to enter, depolarizing the cell. So let's say now we've got dim light. We're moving from the dark to a very dim lit area. Now in dim light, cyclic GMP breaks down. Cyclic GMP breaks down. And when cyclic GMP breaks down, the cation channels are going to close, not allowing sodium and calcium to enter the cell. So let's look at these events in relation to our photoreceptors in relation to dark and light then. So here we can move over to first the dark. So we said in the dark, our cyclic GMP-gated ion channels are going to be open. And this allows cation influx and allowing those photoreceptors to depolarize. So here, as soon as those photoreceptors depolarize, next then you can see voltage-gated calcium channels open in the synaptic terminals. And this is going to then allow neurotransmitter to be released continuously. And the neurotransmitter is going to then cause inhibitory postsynaptic potentials in the bipolar cells. Right? This cell depolarizes, and then it hyperpolarizes the bipolar cell. So hyperpolarization is going to close the voltage-gated calcium channels, inhibiting then neurotransmitters released by the bipolar cell, which then doesn't cause any excitatory postsynaptic potentials to occur on our ganglion cells. Then we'll see no action potential is going to be generated by our ganglion cell, not stimulating then the visual pathway. Then when we talk in light, as soon as we said light strikes, as soon as light strikes, this is going to cause our cyclic GMP-gated channels to close. And when they close, cation influx is going to stop, which then you can see the photoreceptor hyperpolarizes instead of depolarizes, as we saw in the dark. So with this cell hyperpolarizing now, we'll see voltage-gated calcium channels are going to close in the synaptic terminals, not allowing any neurotransmitter to be released. No, the neurotransmitter gets released. So if no neurotransmitter is released, we can see we're going to have a lack of inhibitory postsynaptic potentials in the bipolar cells. So if we're not inhibiting those bipolar cells anymore, they're going to depolarize. When they depolarize, you can see here voltage-gated calcium channels open, causing neurotransmitter to be released. You've seen that whole process. Generating EPSPs on, you can see the ganglion cells, which eventually generate action potentials that will be propagated along the optic nerve to the brain. So you can see both processes here, what happens in the dark and what happens in light. So when we talk information processing in the retina, so as we saw, photoreceptors themselves don't generate action potentials, but we can see that they generate graded potentials, right? EPSPs, excitatory postsynaptic potentials, and IPSPs. So we saw here, light hyperpolarizes the photoreceptors, which then stop releasing an inhibitory neurotransmitter called glutamate, which can be inhibitory or it can be excitatory. And so here we can see then, light is going to hyperpolarize our photoreceptors, which then stop releasing an inhibitory neurotransmitter, glutamate. No longer inhibited, these bipolar cells then depolarize, allowing calcium entry, which allows neurotransmitter release. Now, neurotransmitter gets released onto the ganglion cells, where then it's going to generate EPSPs, excitatory postsynaptic potentials, which then help generate action potentials. So the neurotransmitter released here is also glutamate. So in this synapse, you can see it behaves as an excitatory neurotransmitter, while here it behaved as an inhibitory neurotransmitter. The action potentials are going to be transmitted along this optic nerve to the brain. The synapses between bipolar cells and ganglion cells are not that well understood as the synapses you have here between the photoreceptors and the bipolar cells. So we took care of dark adaptation, light adaptation. Nyctalopia, I want to make sure you got there as well. Night blindness, basically due to rod degeneration, okay, commonly caused by vitamin A deficiency. And then if administered early, vitamin A supplements can help restore function. And then we can see it's also caused by retinitis pigmentosa. So let's check out the visual pathway to the brain. Here then we can move over to this picture. Now here we'll trace the visual pathways to the visual cortex. So we can start with the optic nerve. The optic nerves are going to be formed by the axons of the retinal ganglion cells, and they're going to transmit information to the next area. So here we can see we've got the left eye, we've got the right eye. Here we've got the optic nerve from the right eye, the optic nerve from the left eye. Now you can see some of the fibers from each of the optic nerves are going to stay on their respective side, while others are going to cross over. So you see the same thing happening here with the right eye. You got some of those fibers staying on their respective side, while other fibers are going to cross over. So the crossing over of the fibers is what we call the optic chiasma. So here let's look at our left optic nerve in greater detail, and then we'll look at the right optic nerve in greater detail. Now here we can see some of the fibers, the ipsilateral fibers, they're going to stay on the left side, and they're going to continue back here to the lateral geniculate nucleus of the thalamus. And then from the lateral geniculate nucleus of the thalamus, we can see new fibers are going to make their way all the way to the primary visual cortex, which is found on the occipital lobe. And now here, the fibers that came from the other side, the contralateral fibers, you can see those crossed fibers make their way into the pretectal nucleus and into our superior colliculi before making their way to the lateral geniculate nucleus, and then following the same pathway as the uncrossed fibers. The same thing we have here with the right eye. You can see our ipsilateral fibers making their way to the lateral geniculate nucleus and eventually then sending their signals to the primary visual cortex. And then here we've got our left optic nerve's fibers that crossed over, and you can see here they make their way over to the right pretectal nucleus and the right superior colliculi before they make their way to the right lateral geniculate nucleus. So here we can see the optic nerves will eventually cross over then and become the optic chiasma, which then lead to the optic tracts. The optic tracts. Next, then let's talk depth perception. So when we talk depth perception, we'll come back to this picture here. We can see both of our eyes are placed anteriorly with their visual fields overlapping considerably. From this image here, we can see each eye sees a different view. The visual cortex fuses the slightly different images, providing us with depth perception or 3D vision. It's an ability to locate objects in space. Depth perception depends on the two eyes working together. If only one eye is used, depth perception is lost. Animals, such as birds, have panoramic vision, and their eyes overlap slightly. So let's move over then to vision problems. Now, when we talk problems of refraction, vision problems, first let's look at the focal point of a normal eye, of a normal emmetropic, we can refer to it as well, emmetropic eye. The focal point you can see is going to be right on the retina. It's right on the retina. So first let's check out myopia. Here we've got a myopic eye, or what we call nearsightedness. This occurs when distant objects focus in front of the retina. They focus in front of the retina rather than on it. So here we saw we had to have the focal point on the retina. But here you can see in the myopic eye, the focal point is in front of the retina. It's found in front of the retina. These individuals, they cannot see distant objects. They cannot see distant objects. Myopia results from an eyeball that is too long, or if the cornea has too much curvature. This is basically what I have. We get corrective lenses. Then the corrective lenses are going to help to restore that focal point back onto the retina. Now, if we talk hyperopia, a hyperopic eye or farsightedness, this occurs now when distant objects focus behind the retina. They focus behind the retina. So we correct that with convex lenses as opposed to concave lenses for the myopic eye. So in the hyperopic eye, we use convex lenses, and they're going to help move that focal point forward. Hyperopia results from the eyeball being too short. Results from the eyeball being too short. And then last we have astigmatism. When we talk astigmatism is unequal curvatures in different parts of the cornea or the lens. It's corrected with cylindrically ground lenses or laser procedures. All right, so here now before we take off then and move to the next senses, last thing I want to talk to you guys about here is the results from a Snellen eye exam where you're told your vision is 20/20, 20/40, 20/15, or what you have there. So here when you're talking 20/20, what they're saying there is basically that you could see standing at 20 feet what another individual can see standing at 20 feet. And when you talk 20/40, you're talking about you being able to see at 20 feet what others can see at 40 feet. No, that's not good. You have to stand that much closer to be able to see something versus somebody else. Now, when you talk 20/15, that's good because they're telling you you can stand at 20 feet and see what others have to stand at 15 feet to see. Okay? You can see that at 20 feet. All right, let's move forward then and let's talk about our chemical senses then, and let's jump to smell and taste. Now, when we talk about our chemical senses, we're talking smell and taste. We'll see smell is scientifically known as olfaction, and taste is going to be known as gustation. So we're going to be looking at our chemical senses, olfaction and gustation. So smell and taste. Now, when we go through and we talk about these senses are going to rely on chemoreceptors. They will rely on chemoreceptors. Now, when we talk chemoreceptors are going to be receptors for smell and taste, and they're receptors that respond to chemicals in aqueous solution. They respond to chemicals in aqueous solutions. So what we'll do first is we'll look at the chemical sense, the sense of smell, and when we talk about then the olfactory epithelium, let's move and we'll talk a little bit about this olfactory epithelium. Here you can appreciate that olfactory epithelium. You can see it located high up inside the nasal cavity. You can see overlying the superior nasal concha on both sides of the nasal septum. So now here you can see we're talking location, and then we'll go through and we'll talk structure of this olfactory receptor as well. So here we can see the olfactory nerve is going to be making its way from the cranial cavity down into the nasal cavity. Now we've got olfactory foramina, cribriform foramina, that pierce the cribriform plate of the ethmoid bone. So here we're able to appreciate then the olfactory epithelium, and we'll look at that in greater detail. Now, I want you to know olfaction detects chemicals, odorants. It detects odorants in solutions. Now, the organ of smell is a yellowish patch of pseudostratified epithelium, or we can refer to it as olfactory epithelium, right? Pseudostratified epithelium or olfactory epithelium. And it's again located high up inside the roof of the nasal cavity, covering the superior nasal concha on each side of the nasal septum. That's why sniffing intensifies the sense of smell. You see, because this draws more air superiorly right to that epithelium. The epithelium contains millions of bowling pin-shaped receptor cells. We can also refer to them as olfactory sensory cells. And these cells are surrounded and cushioned by columnar supporting cells. So we'll zoom in on that epithelium. So here, let me orient you to where we are at. Here we've got the cranial cavity, and then here we have the nasal cavity. And then here we can see the cribriform plate of that ethmoid bone. So here we can appreciate the olfactory tract. Then we can appreciate our olfactory receptor cells or our olfactory sensory neurons. These cells, again, we described as being bowling pin-shaped cells. We said this epithelium is made up of millions of bowling pin-shaped receptor cells. And we said these cells are surrounded and cushioned by columnar supporting cells. And here you can see these columnar supporting cells, they're going to make up the majority or the bulk of this membrane. Also found here, you can appreciate basal cells, dynamic stem cells. Some basal cells. The olfactory sensory neurons are bipolar neurons, and each cell we can see has thin apical dendrites. Here you can see a thin apical dendrite, thin apical dendrite, thin apical dendrite. And from these thin apical dendrites, we can see these olfactory sensory neurons are bipolar neurons. And each has thin apical dendrites that are going to terminate as a wide knob-like structure from where we see cilia radiate. From where we see cilia radiate. The olfactory cilia increase the receptive surface area, and we can see they're found lying flat against the epithelium, and they are covered by a thin coat of mucus. This mucus is a solvent. It's a solvent, and it's going to capture and dissolve airborne odorants. This is the only cilia which is non-motile. The axons of the olfactory sensory neurons, you can see gathered. They come together to form the filaments of the olfactory nerve as they pierce through that ethmoid bone and make their way up into the cranial cavity. And here we can appreciate one of the many olfactory glands that are going to be found in the connective tissue that's going to be found supporting the epithelium. So let's move forward then. Here we can see now when we talk sense of smell, humans can distinguish around 10,000 odors. We have about 400 smell genes that are active only in the nose. Each encodes unique receptor proteins. Proteins are going to respond to one or more odors. Each odor binds to several different receptors, and each receptor has one type of receptor protein. So not only do we have these smell receptors inside the nasal cavity, but you'll see also found there will be pain and temperature receptors that will monitor pain and temperature. Next then, let's move to the physiology of smell. So here we can see this nice G protein linked receptor system. Now, when we talk physiology of smell, I'd like you to know for us to smell an odorant, in order for us to smell an odorant, it has to be volatile, meaning it has to be able to vaporize, or we can say it has to be in the gaseous state as it enters the nasal cavity, and it must dissolve in the fluid coating the epithelium. Next, when we talk activation of the olfactory sensory neurons, you'll see dissolved odorants stimulate olfactory sensory neurons by binding to receptor proteins in the olfactory cilium membrane, and then this leading to the opening of cation channels, and then the generation of receptor potentials. If threshold is reached, an action potential is going to be conducted to the first relay station in the olfactory bulb. So here we can see smell transduction. Again, this could be one possible essay there as well. Anything that has a nice long process to it can end up becoming a possible essay, so you kind of will be able to pick up on that. We said phototransduction, then we'll talk about olfaction, and then we'll talk about, and then hearing as another possibility. So when we talk smell transduction, so here we can see everything written, but let's actually visualize everything. So it's the same picture we see here. So here we can see smell transduction. We talk smell transduction. Transduction is going to use a receptor linked to a G protein, we said. We've seen this before. Transduction is going to begin when an odorant binds to a receptor. When an odorant, we can see here, binds to the receptor. This activates G protein. The G protein here is called the olfactory protein. We can see this G protein gets activated and it goes and activates then this enzyme adenylate cyclase. So G protein activates adenylate cyclase. Adenylate cyclase then is going to synthesize cyclic AMP as a second messenger. It will do that by converting ATP to cyclic AMP. So again, cyclic AMP is our second messenger. The cyclic AMP then can act on the plasma membrane cation channels as we see here, causing them to open, allowing sodium and calcium to enter. The influx of sodium leads to depolarization and impulse transmission. Then calcium's influx. Calcium's influx is going to cause the transduction process to adapt, decreasing its response to sustained stimuli. So let's check out then the olfactory pathway. So we'll move back. Here we can appreciate this pathway. So the olfactory sensory neurons, they form the olfactory nerves that synapse in the olfactory bulb. Here you can see, again, you can appreciate those olfactory sensory neurons. They form the olfactory nerves that synapse in the olfactory bulb. So here you can see the bulb. It's the widened out region of that nerve. You can see here you've got the tract of the nerve, and the widened out region is that bulb. Inside of here is where you're seeing the synapse. The synapse is depicted as the glomeruli. You can see it's between the mitral cells and the olfactory sensory neurons. So here we can see the bulbs. The bulbs are going to be the ends of the olfactory tract. The tract then is going to be bundles of axons. You can see the olfactory tract, bundles of axons. Now here we have mitral cells. These mitral cells, they're the output cells. They are second order sensory neurons. The glomeruli then we talked about next. The glomeruli we said are the junctions. And then we've got the tracts, which will be responsible for routing this information then to the brain. So here you can see making its way then to the brain. Let's check out then the sense of taste. Let's move to the sense of taste. So when we talk about taste, I'd like you to know the 10,000 or so taste buds are found on the tongue in papillae, which are peg-like projections on the tongue's mucosa. And some taste buds are also going to be found scattered on the soft palate, our cheeks, our pharynx, the epiglottis, and even the larynx. So you can see primarily the oral cavity is where taste is going to take place. Taste buds are made up of three major epithelial cell types. First, we're going to see here we can go through and we can look at basically one type of papillae. These are circumvallate papillae. And when we look on the side walls of the circumvallate papillae where these taste buds are going to be found located, here we can appreciate one of our taste buds. Our taste buds are going to be made up of a couple of major cell types. Here we can see our gustatory epithelial cells and our basal epithelial cells. Now, these gustatory epithelial cells are going to be the receptor cells for taste. They're the receptor cells for taste. These cells, we can refer to them also as our taste cells, our taste cells. These cells have long microvilli, you can appreciate right here at the end. These cells have long microvilli called gustatory hairs, which project from the tips of these cells and extend through a taste pore to the surface of the epithelium, where they're going to be bathed by saliva. The gustatory hairs are the sensitive portions. That's the receptor membrane for these gustatory epithelial cells. These cells are in close contact with dendrites, you can see here, that are going to represent the initial part of the gustatory pathway that eventually leads to the brain. So going back here, we can see then we have 50 to 100 flask-shaped epithelial cells of two types. Gustatory epithelial cells, the taste cells we said there, and then our basal epithelial cells, which are dynamic stem cells that divide every 7 to 10 days. Next, let's move to basic taste sensations. Now, all taste sensations can be grouped into one of the 5 groups. We have sweet tasting foods, sour tasting foods, salty tasting foods, bitter tasting foods, and the 5th, umami tasting foods. Now, we talk our sweet tasting foods that include sugars, saccharin, alcohol, some amino acids, and some lead salts. And when we talk sour foods, they'll have basically hydrogen ions in solution. Salty foods, metal ions. Bitter foods, alkaloids such as quinine and nicotine or aspirin. And when we talk umami, amino acids, glutamate and aspartate. And then we can see here we have a possible 6th taste. There's growing evidence humans can taste long-chain fatty acids from lipids. And when we talk taste likes and dislikes, they have homeostatic value. They can guide intake of beneficial and potentially harmful substances. Now we talk physiology of taste. For a chemical to be tasted, it must dissolve in saliva, diffuse into a taste pore, and contact gustatory hairs. Now let's look at the activation of taste receptors. I want you to know gustatory epithelial cells contain neurotransmitters. When a food, chemical, or a tastant binds to receptors in the gustatory epithelial cell's membrane, it induces graded depolarizing potentials that cause neurotransmitter release. Then binding of the neurotransmitter to the associated sensory dendrites triggers generator potentials that elicit action potentials in these fibers. And we can see we have different thresholds for activation. We're talking bitter, for example. Bitter receptors are the most sensitive. And all adapt in 3 to 5 seconds. Complete adaptation takes anywhere from 1 to 5 minutes. Let's talk then taste transduction. When we talk taste transduction, I want you to know 3 different mechanisms underlie how we taste. First, when we talk salty taste is going to be due to sodium influx through sodium channels, which directly depolarizes the gustatory epithelial cells. And then number 2 is sour. Sour taste is going to be mediated by protons, the hydrogen ions, which are going to act intracellularly to open channels that allow other cations to enter. And then number 3, we have our bitter, sweet, and umami receptors. Our bitter, sweet, and umami receptors are going to be coupled to G protein called gusducin. We'll see activation. The activation leads to the release of calcium from the intracellular stores, or a blockage of potassium channels occurs, which then causes cation channels in the plasma membrane to open, depolarizing the cell and causing neurotransmitter, which is going to be ATP, to be released. Now, when we talk about the gustatory pathway, we'll see afferent fibers are going to carry taste information from the tongue to the brain primarily by 2 cranial nerve pairs. A branch of the facial nerve, cranial nerve number 7, specifically speaking, the chorda tympani, transmits from the anterior 2/3 of the tongue, and the glossopharyngeal nerve, or cranial nerve number 9, we'll see is going to service the posterior 1/3 of the tongue and the pharynx. Taste impulses from taste buds in the epiglottis and lower pharynx, they're going to be conducted via the vagus nerve. Via the vagus nerve, you can see right back here. So 7, 9, 10. Now we talk taste. So these nerves, then, we can see are going to carry impulses from the taste buds to our solitary nucleus of the medulla. Impulses then travel to the thalamus, and from there, fibers branch to the gustatory cortex in the insula and the hypothalamus and limbic system, where we then have an appreciation of taste. Vagus nerve is going to transmit, we said, from the epiglottis and the lower pharynx. Now, we talk about the role of taste. The role of taste also is going to trigger reflexes involved in digestion, we'll check out later on. It increases secretion of saliva into the mouth, increases secretion of gastric juice into the stomach, and may even initiate protective reactions such as the gagging reflex or reflexive vomiting. Now let's check out the influence of other sensations on taste. We'll see taste is 80% smell. We also have thermoreceptors, mechanoreceptors, and nociceptors that are going to be found in the mouth that also influence taste. Temperature and texture is going to enhance or detract from taste. Next, I'd like you to know a couple of terms. First term is anosmias, which is olfactory disorders. Most result of head injuries and neurological disorders such as Parkinson's disease. And then we have the term uncinate fits. These are olfactory hallucinations. Some patients have olfactory auras prior to epileptic fits. And they'll say, oh, I had this distinctive smell and I know I was going to have a seizure. And then last here, we'll see taste problems are going to be less common. They can happen from infections, head injuries, from chemicals, from medications, or radiation for cancer of the head and neck.